Hello everybody, my name is Claire Gwinnett and I work at Staffordshire University. My Winston Churchill Fellowship was in 2018 and it was very much a hybrid fellowship where it was combining my two main areas of interest, which is in forensic trace evidence and particularly fibres work, and also in environmental science, but particularly looking at microplastic work. Just to give some idea of why I chose that fellowship and where I've sort of come from and why it's led to this, I'll just give you a little bit of background about me. So I've been working in forensic science for over 16 years now, and a lot of the work that I've done has been in sort of competency testing, proficiency testing, quality assurance type procedures, and more recently that has sort of led into doing guidance, consultancy, and doing reviews for ISO accreditation for different police forces. And you might well have seen me in an NPCC working group. Aside from that, my focus has been on trace evidence and particularly fibres, but also hairs, glass and paint. And I've been able to sort of apply that and work with a lot of international forensic providers, a lot of police forces, very much trying to improve trace and fibre recovery, detection and analysis. And that sort of links to the other side of my job, which is sort of the environmental side that I've worked on for less time, so around just under 6 years. And I have sort of developed the environmental work in terms of microplastics at staffs. So I previously did research and casework in animal and wildlife type crime investigations, but I didn't do any sort of particulate analysis for the environment industry. But now, so over the last few years, now I've got the chance to be able to apply my forensic knowledge to the microplastic world, and to do that I've had the opportunity to work with a lot of organisations around the world in helping them set up their microplastic work, and I'm the proud lead of our in-house forensic fibres and microplastic research group that works collaboratively, and it's actually one of the things that have kind of become more formalised and was extended as part of this fellowship. So I'll just talk about that a little bit more later on. So just to give you some context of how and what I do and what I did in my fellowship, I like to sort of think about what I've been working on for quite some years, and that has been in fibres work and trying to understand how things have changed in terms of its use and its popularity over the years. So I like to show this sort of little scribble really, but it's something that I think sort of depicts, especially when I talk and have conversations with other forensic practitioners and police forces about sort of the status play as it was and how it is now. So if we sort of look at this little sort of sketch of a graph, we think of trace work as being kind of a traditional particulate that was well used for many, many years. But then with the advent of DNA, and DNA's ability to be able to answer the who question in a case so very well, then it's given sort of investigators that focus and away from using maybe some of these more traditional forms of evidence. So the focus has gone a little bit more onto the who, rather than maybe also answering the what, the where, the when and the how. These are the questions that trace evidence also answers incredibly well, and we've got a lot of knowledge and data publications that have helped show that over the years. And then we got into this sort of slump because over that time, with the focus on the who, less submissions of fibres over the years, less, and at that point, less knowledge, and it might still be used in major crimes, but we're certainly not seeing it in volume crimes anymore. But I like to think that we're coming out of this now, and this sort of increase, although not steady, is now where we are, partly because of some of the miscarriages of justice that have occurred, where the need to have other evidence and also just not be completely focused on the who as shown to be needed, but also this idea that we can actually now harness technology. We're in the 21st century and we can take trace evidence a step forward. One little sort of warning that everyone's been talking about in terms of trace evidence analyst is sort of highlighted here. So this is Claude Roux, and he made this point in EAFS in 2015, where he was trying to say that this slump that we've had in the use and the popularity of trace evidence in all different cases is actually a really problematic circle, because as soon as we start limiting the use of that and focusing on just one technique, then we're reducing the skill set, the ability to gain the intelligence information we might have been able to get from trace, and then ultimately sort of getting into a hole where we have laboratories that don't have the capacity to be able to analyse and interpret trace evidence. And that is something that we need to break. So a lot of my work prior to my fellowship, and still ongoing now, has been how do we try to break this cycle and improve the amount of forensic fibres work, particularly in the criminal justice system. So I've done a lot of work to try and understand why we've got into that situation where actually trace evidence hasn't been seen as favourably, or you're just getting lower submissions, it's just not used as regularly in casework. So I conducted a survey some years ago with police forces right on the front end of what their perceptions are with the use of trace evidence from scenes, and to try and understand if there was any barriers that were being in place to try and increase their amount of in-house work, or just increase the amount of submissions or use of trace evidence. And if there was any perceptions of the benefits of doing so, because that's important, of course, if you're going to make any major step change, is trying to understand where trace evidence fits within an investigation and the benefits of putting it in an investigation. So these results I had from this sort of survey and these questionnaires and these interviews were not surprising. It's the kind of anecdotal information I'd had for years by working with various organisations. So this certainly wasn't new. But when a lot of these police forces sort of tried to place trace evidence amongst other evidence types, they were putting it, regularly down on number 4, with either fingerprints or DNA first, and then some placed footwear impressions next, and then others put trace in the 3rd place. But nevertheless, they were putting it sort of down there, which I thought is optimistic, and it shows that there's value there. But the kind of points that came out of why actually still it's not going to be used in volume scenes, it's going to be used rarely in other types of cases, although still used in major scenes, partly because of these points on here. So again, things I'd heard before, that it's expensive, and the reason why it's expensive is because there's a large amount of samples, and because of that it's incredibly time-consuming work, not just actually because of the amount of samples, but also the processes that are needed to search for that evidence and then prepare them ready for future analysis, so the actual dissection of them away from a tape. And that, along with the need for experienced analysts and lots of expensive equipment, so our perception that is the thing that's driving up the cost. With that, the main idea that came out was, if you make this more cost effective, so we maintain the value and we're able to provide this information, so we need more data, we need to be able to use this for activity level information as well as using it for source, but trying to lower the costs at the same time, that would be what would lead to the increase in fibres work. So over the years, we sort of focused on trying to maximise some of these benefits, and some of these benefits were seen in this survey. I'm just flagging them up here now because this is not just me saying this. This also came out of these discussions with police forces, that actually fibres and other trace as well, but thinking about fibres now, that we're regularly going to see them at scenes where we may not necessarily see DNA or fingerprints. They've got a great ability to be able to provide intelligence information. They're great for linking between scenes, between individuals, and they're seen as really valuable for multiple scene analysis, so linking between scenes. And some examples that were brought up within house burglaries, with prolific offenders who don't leave these other evidence types, that fibres evidence will be present, but we need a mechanism to be able to maximise their use in these cases. So this is a lot of the work that I've sort of done, is trying to focus on, okay, how can I try to do that? So I focused a lot on decreasing the analysis time. So knowing from experience and talking to practitioners that there's a large amount of time taken in the actual searching for traces. whether that's fibres or other traces. So we have to search a tape, try to identify what's of interest, and then we need to extract it in order to be able to go and move that onto polarized light microscopy, Fourier transform, infrared spectroscopy, and so on. So we've got to dissect it out of that tape. So that is incredibly time consuming. It's also subjective, and it's also a possibility for loss and contamination of the sample, which is other issues there. So I'd focused my time a lot on trying to get a system that allowed the recovery of these fibres and other traces, and allowed them so that they didn't have to be dissected away from the tape. And I moved from prototype work with a tape, and then with my colleague Professor Andrew Jackson, we got to this stage where we've got a tape called EasyLift that allows complete in situ analysis. So it removes the need for dissection, so it reduces that time consuming step, and it allows in situ analysis for all different types of method. So for example, we talked a lot with practitioners around what methods they particularly used and what their workflow is as standard with fibres. And we tried to sort of see whether actually that could be done in situ on this tape. And these are just some of the collaborators that we worked with who gave a lot of feedback based on this tape and whether this would decrease analysis time. And the result of that is a tape that allows comparable analysis of whether this was mounted, for example, for polarized light microscopy, whether this was mounted in a normal mounting medium or whether it's in the tape. It is optically transparent. It causes no interference for confocal Raman spectroscopy. You can do fluorescence microscopy because it's non-fluorescent, and you can do microspectrophotometry. So it's something that is removing the need for dissection for at least those techniques. But unfortunately, that doesn't solve the main problem here. That might reduce the time needed in that step of the workflow, but it doesn't remove the need for searching for those traces, and it certainly doesn't do the analysis automatically. So there is a need here to get a full system that allows this automated analysis of these traces, something that has been thought about, attempted over the years, but at the moment we do not have an automated system that detects, that analyses, and then the added bonus helps interpret what that means in terms of its evidential value. And that is where my work is really going, and that's kind of what has come out of my Winston Churchill Fellowship. Now I'm just going to take a segue now of how this has come together, because I didn't go around countries and ask them how they were trying to develop automated systems for forensic fibres work. In fact, I investigated a completely different sector, and that was the environmental sector and those who were interested in working in reducing microplastics, detecting microplastics, and trying to understand their abundance and source in different environments. So something that seems very, very different, but actually there's some clear similarities, which you may already sort of, you can think or guess. I got into microplastics wasn't just this fellowship. I'd actually started working in this in around 2015, where I got invited to contribute to a project along with Oxford University on the analysis of some microplastics that had been found in the deep sea. Now, their sampling techniques are very different to what we would use at crime scenes, and they are going out in big boats, they're using ROVs to gather sediment and also gather organisms to see whether they've ingested and to see whether there's the presence of microplastics there. Now, a lot of microplastics that are present in the environment, we now know are actually fibres. So they're microfibres, which is, of course, a main focus area of what I do previously, but for the forensic sector. So for this work, it was really about trying to understand what the level of characterization could be applied to these little single fibres that are being found attached and ingested in these deep sea organisms, so as to better the information that was currently being published, which was as basic as saying it's a fibre that's blue, that's nylon. And as we know from the forensic science sector, we say a lot more than that. We characterize its optical, its morphological, its chemical properties, and at the same time we quantify the color, don't we? So it's applying some of that to see whether we can get more value out of what we find out from microplastics in these environments. At the same time, this study highlighted the need for something else. And led by Lucy Woodall, and she was at the Natural History Museum at the time, the question that was posed was that when we've got such small amounts of fibres found in the deep sea, or wherever, but in this case the deep sea, how do we show that it wasn't contamination during the recovery and then the processing? And this is where I started to work to show how forensic science protocols that we use to minimise contamination and also to monitor it can be applied in this particular type of study. And that's what we've got here. So this was a study that was published that has actually led the way into a lot of processes that are now used in microplastic work. And it allowed the chance to be able to show how forensic science and the things that we do can be applied in this particular discipline. Now, through those years, I've had to say a lot of the time, like, what am I doing as a forensic scientist doing in plastic pollution? I have to explain what forensic fibre analysis is, and then with the end outcome of sort of saying polymer analysis is just polymer analysis, right? That's what this is all about. Whether you are analysing these fibres from the deep sea or whether you're analysing them from a crime scene, we have got benefits by using some standardised, robust approaches that allows us to maximise our knowledge about the sample. And these overlaps that I sort of saw early on with forensic fibres work and the knowledge we've gathered over many, many years, and what that can benefit the microplastic sort of industry and researchers. So we're kind of could be summarised in these points here. And I'm not going to go into these, but what became sort of a bit of a question as I sort of developed working with various microplastic researchers and organisations was that actually, is there anything that we can learn from any of these points, but maybe particularly this one here around faster and more effective quantification and characterisation? Is there anything going on right now, knowing the thrust for new knowledge in microplastics? Is there anything that we could borrow and use as forensic fibre analysts? And that's led to my fellowship, where I see this as a hybrid really. I've come from a forensic fibres knowledge background, and I've wanted to show that we can apply that knowledge to microplastics, and there's benefits there. But my fellowship is about really where these two overlap. So in the middle here, which we might call environmental forensics, but actually microplastics is not a traditional subcategory of environmental forensics. So as much as it is for me, but for many individuals it's not. But I'm kind of trying to make it a category in this particular fellowship. So from that, this was my main aim, original aim, where I was interested in going out, talking to eminent scientists that worked in microplastics to find out what are they doing, what technology are they harnessing in order to improve their analysis. And I was quite early on, this got adapted to actually two things now. Where can I knowledge share to these individuals about what I do in forensic science to benefit their work in microplastics, but then at the same time, actually, where are they going, especially where do they see themselves going by their horizon scanning of the need for certain knowledge in microplastics, and how can that help us now or in the future with our forensic trace work? So this was really interesting discussions, and I got the chance to travel to two countries. So I went to the US first, both East Coast and West Coast, and I was trying to harness knowledge and expertise from a whole breadth of different individuals. Those that are researchers who are trying to quantify and characterize microplastics, those who are trying to use the data generated to inform policy, or those that are the leaders in developing what the next steps are, so funding bodies. So I visited Washington and the NOAA, who actually have a marine debris program that is about the next steps and moving forward in microplastic work. And I did the same in Australia again to get these two countries who are very forward thinking in microplastics and have developed a lot of techniques, tested techniques, and tried to drive this forward in a very short period of time. So what was the key findings? I chatted to all these people, I got to shadow what they did, I got to see the techniques that they used, and what was prominent was the fact that microplastic studies, the approaches that they're using, the knowledge that they're gaining is It's very, very, very fast moving, by far faster moving than what we have had in forensic trace work over the years. We've had the passion, we've had the expertise, but we might not necessarily have had the funds to be able to drive this forward as fast as we could, whereas in microplastic world, they absolutely have. So because of that, they have been able to think about greater sensitivity techniques, so that microplastic researchers now are not just interested in the micro scale, they're interested in the nano scale and what that means in terms of detection and analysis. And at the same time, this idea of generating large amounts of data in order to be able to understand what they are, what they're seeing, and the abundance of those and the potential sources of these microplastics was really key. Something actually that has only developed over the last few years, but has rapidly took traction and is now getting some real key outputs. At the same time, in the other part of the sector of those industries that are creating new equipment, they're really keen to sell this to microplastic researchers. So the idea of automated or semi-automated, or the use of AI for the detection of these particulates and then their subsequent analysis is huge. They're pushing that, they're selling that, because they actually see that as a greater commercial profit than it would be if you were trying to sell it to the forensic sector. Having said all of that, they still have a lot to learn. That was one thing that picked up. There's still a place for us to share that knowledge from forensic science to the microplastic sort of sector, especially in contamination prevention, and especially in the ability to be able to use a suite of different approaches, methods that can fully characterize a fibre. So again, looking at its morphological, optical and chemical properties rather than just focusing on one or two of those. And they've got the same drives. That's what really came out of some of my findings, was that more data, but also wants the analysis to be quicker and cheaper. So it's getting that value out of money, because a lot of these studies, they initially started off small scale, but now we want mass scale, surveys of water, of land, of air, that's constant monitoring for these pollutants. And to be able to do that, it has to be fast and cheap analysis. So that we also have in common the types of questions they're asking of that. So just to repeat some of the points I've already made, they're interested, as are we, is what's there and how much is present, and where's it come from, so its source. They're interested in where it's going, so in that environment, but we're kind of interested in that, and also from the point of view of persistence, in how long it's been there, and then where is it going. So we've got some similar, although maybe not exactly the same questions, but it means that we're trying to drive towards the same innovations to try and answer them, which is excellent. Because what that means is our attempts at automation over the years, which are superb, can combine forces with their approaches for automation in order to be able to get a complete system. So just to show you here, and this is just a little overview of some examples of how we've progressed as in forensic science for fibre detection and analysis from an automated point of view over the years. And we know back in the 1980s, we had fibre finders created, the MaxCam, the FX5, that had some inherent problems, although the idea and trying to harness the technology that was present at that time, was excellent, although it's not fit for purpose. So there was problems with false positives and the hardware jammed. So it's come on from that. And there's been multiple studies, not lots and not necessarily regularly, but those that have looked at trying to use the likes of colour space models, so image processing models using 3 layers of information from the RGB values. There's been studies that have looked at trying to count and detect traces, like particularly fluorescent fibre, so looking for that characteristic. Where I see this is going, though, is in the realm of machine learning. And how do we use AI, along with our knowledge of what is required for our detection and analysis of fibres? How can we build the system that allows for that? Now, microplastics aren't far behind in that sense, and in fact their continued use of automated systems or semi-automated, as I've seen across the world really. So a standard use of a semi-automated Raman spectroscopy in order to be able to get chemical information but also morphological information at the same time, that's regularly used now in microplastic studies, as is Spotlight 400, which is using an imaging system for the detection of these particulates and then gathering that spectral information. So the uses of AI are kind of coming off the back of that. These don't necessarily use AI as we might think of it, but we are seeing areas such as plastic tide that is actually harnessing AI algorithms for macroplastics. So it's being applied to macro, so we can see it might go to micro next. Now, from that has led to some of the work that I've done, and you probably might be surprised, what with all the build-up, that a lot of the work that's come out of my fellowship has been around trying to apply forensic techniques, and that I had the fabulous opportunity of doing on an expedition last year with 2 of my PhD students that was monitoring microplastics along the whole length of the Hudson River, using the like of EasyLift for the recovery of these particulates. Part of that was around trying to gather training data sets for our own automated system. So I'll just tell you about that, but one of the other things that came out of this fellowship was the formalisation of the research group here at Staffs. We now are multidisciplinary. It is not just from a forensic science point of view, but from all these disciplines that you see in here. So it's sort of, again, harnessing this expertise both in computer vision, data analytics, evidence evaluation to try and solve these problems or improve methods for both forensic fibre work, but also microplastic work at the same time. So it's actually a research group that's bringing these two things together. And from that, we've got many collaborations. The fellowship kicked this off, but actually we've managed to gather this not just over the years, but since then with this work, and now we have collaborations in 22 countries. And from that, we're really continuing the learning of everything that sort of just happened in those first 2 visits. Part of that learning has led into this automated system. So this vision that was there, that I wanted to be able to use artificial intelligence in order to be able to create a system that allowed the detection, the identification and characterization of microfibers, and allow that to form a database that could form conclusions are based on prevalence, abundance, and also be used in the likes of the Bayesian approach. So this has actually sort of come out of the work from the fellowship. This has got a PhD project at the moment, and Amy Osborne, who is developing the training data set, using some of the things that I've learned from my fellowship to try and create this system. And I'm just going to focus on this micro one here because we're also interested in generating an AI system for the macro detection of these pollutants. So you'll see here, this is very much around the plastic pollution point of view, but you can see that this overlaps with a lot of the work that we want to do in fibres work, forensic fibres work. And this is something that I've discussed with a colleague of mine who developed Spectral 360, which is a machine learning system that ultimately overcomes some of the problems with other sort of machine learning algorithms. So our idea is that we develop the ability to be able to image under different light sources, and it won't matter if there's variation in those light sources between microscope to microscope. So whether I'm analysing microplastics at Staffs or someone's analysing them over in Australia, it doesn't matter the background of those fibres on the tapes. It doesn't matter whether it's heavily debris. It doesn't matter what lighting setting they've got that on in terms of amount of illumination. It can handle that because this system here is bio-based approach. So it's there. It very much mimics the brain and the human eye so that it can adapt to these different illumination changes and what we call camouflage. So the things that might actually interfere with our detection. This is the system that we're using to build an automated analysis system for microplastics. But of course you're thinking, well, I'm interested in microplastics maybe, but how does this to forensic work? Well, our training data set here is predominantly fibres, and this will be, so we're using that knowledge in terms of how we can get spectral information from these fibres, and all that knowledge that I've sort of gathered, I'm feeding a separate project which is that is based from the Shuttle Consortium, which you may or may have not heard of. And I'll just end on this little bit here just to show where we might be going, and hopefully we'll be going for trace work in the future. So this project was a consortium that bid for Horizon 2020 to actually fulfill this vision here, which is an automated system that allows the recovery of traces from scenes, all different traces, in fact, not just fibres, but including skin cells, gunshot residues, hairs, paint, etc. And then allow that to be automatically scanned. And these recovery system is to be a tape, and it should be able to automatically scan, detect these different particulates, analyze them in situ for a whole range of different characteristics, which will then feed into a database that then can facilitate some data analysis and allow us to be able to inquire its provenance, its prevalence, whether this links between scenes, and gather that intelligence and comparison information that we need. Massively reducing the amount of time because it would be automated, at the same time allowing us to be able to get large amounts of data. So the system's just being built right now, and I've got the pleasure of being part of this project as an employee of Spectracon, and it's those with Zanassi and partners who are building these 4 tools. So a tape lifting system that you can use on all different crime scene surfaces, and they can be all the way up to A4 in size, and allows in situ analysis, that feeds into a lot of the work I've done previously. And then that is going to be placed on a microscope system where it can be analysed under a variety of different light sources, and to characterise those traces by using machine learning algorithms through image processing in order to be able to do the detection, the quantification and the characterisation. All of that information is to be fed into a database, and that database is to be used by both front end, so with police forces who want to use this for triage and try to understand what the evidential value of a trace might be to order for them to get a more effective strategy for their investigation, but also for forensic practitioners to utilise this database to understand prevalence, to give some idea of provenance of these samples, and allow some pattern recognition. So a sample that's turned up on a shuttle microscope in one country might end up being compared to a fibre, a trace that has turned up on another shuttle microscope, that with this shared database approach. And at the same time, is to give us that opportunity as forensic scientists to actually have a chance of having these large data sets, things that allow us to understand transfer and persistence a little quicker, and to think about how fibres might be, or other traces might be distributed in a given case scenario. So for me, the future is very bright from after from my fellowship, and I'm very keen on seeing where this goes next. If you're interested in sort of collaborating or getting in contact, please do. And for now, thank you for listening, and I hope you enjoyed that presentation.